Hi guys, today I discuss the topic gene manipulation and commercial utilization. My content is Introduction, Techniques of Genetic Engineering, Commercial Utilization, Advantage and Disadvantage and Conclusion. And start introduction. Artificially copying a piece of DNA from one organism and joining this copy of DNA into the DNA of another organism. It allows gene from one organism to be inserted into a cell of a different organism of a different species, and it is a GMO, and genetic engineering or genetic modification, another name. Next example, human genes can be inserted into a bacterium. Next is animal genes can be inserted into cells of other animal. Third is plant genes can be inserted into plant cells. Techniques of genetic engineering. There are three techniques: recombination DNA, PCR, cell electrophoresis. And recombination, the ability to combine the DNA of one organism with the DNA of another organism. And recombination DNA technology was first used in 1970s with bacteria. Next is summary of steps of recombination DNA. On this picture has the summary of steps of recombination DNA. Next is summary of steps. Remove bacterial DNA. Next, cut the bacterial DNA with restriction enzyme. Then cut the DNA from another organism with restriction enzymes. Combine the cut pieces of DNA together with an enzymes and insert them into the bacteria. Next, reproduce the recombinant bacteria, and the foreign genes will be expressed in recombinant bacteria. Next, PCR. PCR stands for polymerase chain reaction, and this technique that can produce over a million fold amplification of target DNA within a few hours. And the PCR technique follows these steps: denaturation, annealing, and elongation. This diagram is a polymerase chain reaction. Next is denaturation. When the double-stranded template DNA is heated to separate it into two single strands. Next is annealing. When the temperature is lowered to enable the DNA primers to attach to the template DNA. Extension. When the temperature is raised and the new strands of DNA is made by the tag polymerase enzymes. Next is gel electrophoresis. It is a laboratory method used to separate mixed DNA, RNA, or protein according to molecular size. In gel electrophoresis, the molecules to be separated are fused by an electrical field through a gel that contains small pores. Next is commercial utilization. In medicine, mass produce insulin, human growth hormone, etc. In agriculture field, increase crop yield, in reduce cost for food or drug production, and transgenic animals. Next is some transgenic animals: chicken, more resistance to infections; cow, increase milk supply and learner meat. Next is goat, sheep, pig, etc. Next is advantage and disadvantage. Advantages are can be used to make protein from other organisms on a large scale. Next is can be used to produce medicines and can be used to produce organisms to specific characteristics. Disadvantages: still new, may be unknown long-term side effect, and gene manipulation curve can mix with the wild population and residue variety. Reduce variety. Next is conclusion. Genetically engineered crop can reduce some environmental risks associated with conventional agriculture, but will also introduce new challenges that must be addressed. And these are my reference. Thank you. And मेरा ये वीडियो अच्छा लगे तो मतलब समझ में आया है तो मेरे को एक लाइक कर दीजिएगा। थैंक यू मेरा ये वीडियो देखने के लिए।